Okay, thanks everybody. Welcome to another edition of Conversations with Dr. Cowan and Friends. And I'm actually looking forward to this one. I didn't know Marvin Haberland, I think is how you say it.
Yes.
Until maybe 3 or 4 or 5, I don't know, days ago, when somebody sent me an interview that you did with Sam Bailey describing your court case. And I'll let you describe that in a minute and a little bit about who you are. And I watched the interview and I immediately thought, I would love to talk to you to do something that I've been waiting 3 years to do, which is I've said to in many situations, the last person that I would want to question me about virology is me. Although I also wouldn't appreciate being questioned by Andy or Stefan or Mark Bailey, because I'm sure they can think of some tough questions. But I was so impressed with your how much you've really taken this on that I thought, I wonder if you, Marvin, would be willing to be questioned by me. Because up till now, I haven't been willing to tell people what the questions are. And interestingly, you readily agreed. And then I asked you if you wanted to see the questions beforehand or you were willing to do it blindly. And you said, I'll just do it blindly. And that also convinced me that you're confident that you know what you're talking about, which I already knew. And there's 2 other reasons why I thought this would be a good idea. One is, as we'll hear, in this court case, at some point you may be cross-examined. And so it might help to hear somebody who's trying to cross-examine you and isn't this right? So it may not be the first time. So I thought that might be helpful. And the second thing is, I know it's not really time yet, but at some point you may need funds for legal and helping with that. And I think we can do that. So we may not need to do it now, but at some point we're gonna, if need be, we'll help whatever you need with the funds for legal or court fees or whatever it is. So those were my 2 main reasons for doing this. And I think there'll be a lot of people who are interested in what you have to say. So with that, Marvin, just tell us pretty briefly, because people can watch the interview with Sam, just, you know, who you are and what it is that you're doing with this court situation. And then we'll go into the questions about virology. And welcome.
All right.
Thank you.
Yeah, thanks, Tom, for the invitation and this nice introduction. Yeah, I'm Marvin. I'm 30 years old and I'm from Hamburg, Germany. And basically, I'm working as an engineer, and I discovered the field of health, nutrition, virology when my grandmother passed away, and this from cancer therapy, and this really got me into the topic. I did a lot of research and investigation and found the work of Stefan and many others that was very interesting to me, because it felt kind of right to me. The claims that they were making were very profound and scientifically very stable, and I was able to verify what they were saying, and that I was reading through the papers myself and verifying that that the proof for a virus was not in these publications. So what I did when COVID started is basically when Germany introduced the measures like wearing masks in the streets or other measures, I did not follow the measure to get a fine, because in Germany this opens you the door to go to the court, right? If you get the fine for not wearing a mask, you can then disagree and send it to the court, which I did, following Stefan's advice to do that actually. And then I now received the invitation for a court case. However, It should have been on the 19th of October, but the judge is, apparently ill or sick, and I received a letter from the court of Hamburg that the case is like this appointment is canceled. So far, I haven't received a new appointment, but I'm pretty sure I will hear from them soon. Let's see. I hope for the best. Basically, the viewers can check the interview with Sam, but I can very briefly say what is the topic of the case. So I'm saying that the measures in Germany are based on the law of infectious disease, and the law of infectious disease in Germany states already in its first paragraph that every virologist, every university, every authority has to follow the scientific rules, that everything which is based on this law has to be supported by the status quo of the scientific method and scientific technique. And I demonstrate in front of the court that this is not the case in virology, because there are no scientific controls documented and no scientific controls performed in the publications, which is already enough to show that this is not the status quo of science. Because in the exact sciences, it's very important to perform scientific controls. Of course, no one would disagree with that.
Yeah. Okay. And so that's what makes this different, is everybody else is suing like, well, these are too harsh, or it's stomping on my constitutional rights or whatever. But this is really what we've been saying, the heart of the matter, that there is no virus and proven to exist. And that's because you, the virologist, have failed to do controls. So therefore, the whole thing is invalid.
Yeah, you say it correctly, because if you go to the court and say, I don't know, measure A or B or C or the vaccine or whatever is against my human rights, yes, okay, but the court can always find argumentation, or always say that if they balance the health of the society against the individual right, then okay, they can always find an argument. But by pointing to that core question, this fundamental question, they cannot escape so easily. And this is really pinpointing then on the law of infectious disease, because it states that it has to be supported by the latest status quo of science.
Yeah.
And this is basically a different approach that I'm doing than all the others before me.
Right, which is exactly the approach that I and others have advocated from the get-go. So
Yes.
Let's now look at something just to start us off. And this I received actually just today. You may have seen this, but this was an article. Now, let me just be clear about this. This is not in a peer-reviewed journal, and some people may quibble about that. But for me, peer-reviewed is sort of like the old boys network. And so, of course, anything that's actually honest and realistic is likely not going to get peer-reviewed. And so that's like a catch-22. They say, well, you can't. It isn't in a peer-reviewed journal, therefore I'm not going to pay attention to it. And that's because they won't let it in a peer-reviewed journal. So it's like heads I win and tails you lose. But
yeah,
this was
Tom. There is an interesting point, because there is a German court decision which actually says that science is not bound to universities or to peer-reviewed journals exclusively. Science is not defined only by those entities, but scientific method and science can be performed rather by everyone who follows the method. So there is a clear statement in Germany for that.
That's great to know. The other problem with this is the author is not identified by name. It's head of an independent laboratory in Germany. And one could say, well, that automatically invalidates the study. But given the climate and the fact that whoever this laboratory or person is, this study may create problems for them. I think that everybody can see that that is not a ridiculous thing for this person to do, to not identify themselves by name. I mean, we all wish it wasn't like that, but that is the case. Now, I just want to get...
It's also not relevant for the content, for the scientific argument, right? It doesn't matter if it's a grandmother, if it's I don't know, a politician, whatever. It only matters what is the argument.
Right, or Pumpkin the cat. If he did it, that's fine. So here's the experiment. You know, on behalf of Stefan, we checked whether agents other than, I'm reading the experiment, other than alleged measles virus can lead to cell fusion with resulting death is equal to syncytia formation, otherwise known as cytopathic effect. And the cell cultures that look exactly like the one based on the publication by Enders in 54. For this reason, work was carried out in accordance with the protocol of the World Health Organization for the detection of measles infections in cell cultures. And then they give an exact description of what they did, and I'll let people read this on their own. And then you see the results, which were depending on the non-viral and non-infection substances added, changes in cell morphology could be observed at different times, which since 1954 is always equated with isolation of the measles virus. In other words, depending on what you add, non-viral or viral, I would even submit they didn't know that they added anything viral, you get different observations, particularly after the addition of high concentration of penicillin, streptomycin, or cultivation under deficient conditions, the 1% fetal calf serum. Changes in cell morphology were found that were microscopically identical to the syncytia formation described as the measles virus. In other words, this which is given as the proof of the measles virus, this person is saying that this is not the case. This can be because of the other conditions of the culture. And these studies have clearly shown that syncytia is not specific for measles infection. In other words, what we use as the proof turns out not to be the proof. And thus the forgotten observations of names here that proving the existence of a virus with this technique were actually only assumptions. In other words, they weren't proofs at all. So this puts a big dent in the idea that cell cultures are the proof of the isolation of the virus. So I just wanted to put that out in front. And now let's get to this study, this rebuttal to me by Jeremy Hammond that said that I am saying things that are untrue. Let's start with, he gives a definition of isolation here. Yes, we can define what virus isolation means to scientists. It means to separate the virus from the host in a way that enables scientists to characterize and identify the virus. The gold standard for doing this is the cell culture. And by the way, one of the reasons I wanted to have Marvin is not a virologist, not even a medical person. And I would say it doesn't matter, because it's just about whether you can read the studies and think. So Marvin, what's wrong with that definition, or is that a correct definition of what isolation means?
Well, I would say that the first part, scientists to purify the sample before doing cell culture, there is already the mistake, because they don't need to purify it before doing the cell culture. It is important to purify it after the cell culture, because the virus, as they say, is grown and multiplied in the cell culture. So we want to purify it afterwards. It is true that they purify sometimes samples before culturing them, but it doesn't matter. We want to prove the virus. We want to obtain the virus separated from everything else in the cell culture. That's why we need to purify it afterwards. And this they never do. And this is the point. This is already debunking the whole thing. I mean, what he says, it doesn't even matter.
But just to clarify, when you say that they purify before doing the cell culture, that also, I think, depends on what you mean by purification, because to most people, that implies if you purify before the cell culture, then you have pure virus before
No.
that's not the case.
No, what he is saying is that purification beforehand, the sample, they just run it through a filter or they run it through a centrifuge sometimes, but they will not assess the result. They will not check the result with electron microscope. They will not perform experiments like transmission experiments with that and so on. They will not do any further experiments to check if there is a virus inside this purification. So they just will take it, put it in culture, and from there on they will go on. So we need to purify the final result of the cell culture or any result, and then prove that this purified virus or particle caused disease. This is the point. We need to, at what point ever the purification is done, immediately after that step, you will need to do transmission experiments with that purified sample, and you would need to prove that it is a purified sample. There is no electron micrograph from any of these purified samples at all. So this is the point.
Yeah. So you're not aware of any studies that takes a material from a patient and purifies it to the point where they show you only purified virus before the cell culture?
Well, some show purified particles, but they haven't proved that they are viral. Just showing a photograph doesn't prove anything. You need to prove that these particles that you are showing are actually causing disease. So you need to take them out and you need to, spray them in the air of monkeys or of cats, of dogs, of humans, whatever, or put it in the food. And you need to prove that then you cause the same symptoms in those animals compared to a control group where you spray just water vapor or whatever. And then you could say, okay, yeah, these particles on the picture are actually viral once you showed that they cause disease. But just showing the picture doesn't prove anything. So
yeah,
it doesn't matter.
I would actually change that a little bit and say, I'm not aware of any study that showed that even looked at the electron microscope band of a so-called virus before the culture.
That's correct. That's the same. That's also my knowledge. Yeah, that's correct.
There is no. So to say that's to be technically correct, there is some purification steps before the culture, but there's no evidence of a purified virus. I think that's the technically correct way to say it.
Yes, exactly. You could actually run centrifugation by sucrose density gradient beforehand with the sample. You could do that, but you could even show electron micrographs of that result. But it still doesn't prove a virus because you also need to show that it is pathogenic. So you need to run further experiments with animals, whatever. And but you're right, there is not even a photograph of a purified sample through sucrose gradient. So I'm not aware of any studies that do that.
Right. I don't think there are. So let's get into the main study that he talks about. No, that's not it.
Yeah, wait a second. Go back, please, Tom, because here I would also like to point something. Here he says the bold text. The premises. So he says the premises of your questions are false. It isn't true that scientists claim that SARS-CoV-2 exists solely based on the finding of pieces of the virus. It is not true that they never purified a sample prior to inoculation in the cell culture, okay? It is not true that they have only sequenced pieces of the SARS-CoV-2 genome. They have sequenced its whole genome, which independently from the isolation of the virus in cell culture is proof of the virus existence. And this is not correct. They have not sequenced the whole genome. They have sequenced the whole cell culture. But from there, they had to use computer programs, namely Trinity or Megahit or all the others, and they had to calculate the whole genome. No one actually sequenced the whole genome. They sequenced the whole cell culture, the whole mess. And because they don't know what parts are apparently viral or not, like supposedly viral or not, they have to calculate that. And then they get gaps in those results. And then they need to use so-called gap-filling algorithms to create the whole genome. So what the guy is saying here is totally wrong.
In other words, what you're saying, interestingly, I think the word sequence is the wrong word for what they're doing. They're creating, they're aligning it or assembling a genome, not sequencing a genome.
Yeah,
So,
they are sequencing the cell culture. That is the correct.
They're sequencing the RNA or the DNA from a cell culture.
Yes.
Not from any particle called a virus.
Exactly. And the only reason why they need to run the computer programs for getting the whole genome is because they have not obtained the virus in a purified state. Otherwise, if they had the virus in a purified state, separated from everything else, then they could sequence the virus in a whole approach without using any tools like metagenomic sequencing. They wouldn't need to do that. They could just sequence it with gel electrophoresis, whatever. But because they don't have the virus, they are using the computer algorithms.
Right. And they never find, even then, correct me if I'm wrong, do they find the whole genome when they do the assembly or not?
Well, they create a genome and then they would need to prove that this whole genome actually exists. And secondly, they would need to prove that it caused disease.
Yeah.
So only to calculate something is not proof, first of all, that it even exists, and second of all, that it causes disease. It could exist, why not? But then you show me, please, that it causes also disease, and that they never do.
Got it. Okay, let's get to this study. I think it's here. Yeah. No. That's not the right study. I should have. Yes, here we go. Mm-hmm. Okay, we're going to look at this is one of the main studies that he uses. So here we are with the and one of your main claims is they don't do controls when they do these experiments. Yeah. So here they have 4 lower respiratory tract samples, including bronchial alveolar lavage fluid collected from pneumonia. 7 were collected from patients in Beijing with pneumonia of known cause and these serve as control samples. Extraction of nucleic acids from clinical samples, including uninfected cultures that serve as negative controls, was performed by all this and tested for viruses and bacteria using PCR, and that's the program. So why isn't this a valid control?
Yeah, if you actually download the whole publication, they even show a picture of that, what they call mock infection. So why is it not a correct scientific control? If you go to the supplementary part, I think if you scroll down the whole page, you will, supplementary, exactly. Here you will find the methods part. Here, supplement material. Go down, scroll down further. No, this is not the right document. There is the methods. There is a supplementary for methods only. I think it's, if you scroll down this page, like at the bottom of the publication, if I remember correctly, supplementary material, supplementary appendix. This is, yeah. Is it the same?
Yeah, it's the same.
Okay, let me. So anyway, if you find the correct methods, because they are not given in this publication, the whole methods section,
right?
There you can see what they did in the methods part, and they only explain for the standard procedure what they did, but they don't document the scientific control. They will not tell you what exactly they did for the mock infection. And mock infection is not a defined, self-explanatory statement, because mock infection could mean that you have a cell culture by itself, you add nothing, you just leave it there. Mock infection could mean you have a cell culture and you add the same ingredients and the same antibiotics and everything the same. Or mock infection could mean that you have a cell culture and you add slightly different chemicals to it. We don't know. We don't know at all, and we don't even know what they add to that cell culture in terms of samples. Do they add samples? Do they add samples from healthy people? Do they add samples from sick people with other symptoms? Do they add no samples at all? We don't know all of that. And in science, for the scientific method, it is important that you document all the steps, because other scientists need to verify and validate and replicate, be able to replicate the experiment in the future. So just saying in the publication that you had done a mock control is not enough. You need to document. This is scientific rule. You need to document what did you do in that mock. What is the mock control? What is it made of? What are the steps? And this is not documented in the whole publication.
Right. Interestingly, here actually is I got from Science Direct, a definition of what a mock infected is supposed to be. Control used in infection experiments, 2 specimens used, one that is infected with the virus of interest, and the other is treated in the same way except without the virus, sometimes a non-virulent strain. So this is what we're meant to believe is a mock infection, which is not, I would say, just a science definition. It's also the definition from common sense, right?
Exactly. Yeah, exactly.
So you have one experiment where you have cell culture and you put the whatever the snot or the lavage that you think has the virus. And in the next culture, every single thing is the same. The nutrients the same, the antibiotics the same, the fetal calf serum is the same. Everything is except you took the virus out of the sample.
Exactly. And this, of course, they haven't done, because that would mean that they purified the virus, and they haven't done it. So in this case, it could even mean that the mock infected cell culture has no sample at all. And that would not be a good control, because all the other sequences inside the patient sample would be missing. It would not just be the independent variable, which would definitely define the good control, but all the other variables in the sample, in the human samples, would be missing as well. So it is not even if they did it like this, it would not be appropriate.
Right. So let me just flesh that out for a minute. What you're saying is, so first of all, if you're going to do a cell culture with a sample of lavage fluid from a sick person, which allegedly has the virus, nobody would claim that the only thing in there is the virus, right? There's
Correct.
proteins and dead cells and enzymes and nucleic acid pieces, et cetera. So you have many variables in there. So if you just in the mock infection don't put a sample from a sick person, or even if you put a sample from a healthy person, you have no way of knowing which of those variables created the cytopathic effect or will show up in the final assembly of the genome.
Correct.
That's what you're saying?
Yes.
Yeah. And as far as we can see, this paper certainly didn't take the virus out of the sample.
Correct.
And in fact, we don't actually know what they did because they didn't say it, which means this by definition can't be reproduced. Therefore, it can't be science.
Fully correct.
Okay. So I think it's clear this is not an example of appropriate controls. And I would just point out, just because you use the word control doesn't mean it's true, right? You can say a lot of things, but that's not what science is about. Okay.
That's right.
All right, here's the next thing. But Marvin, we did electron microscope pictures. We have them right here. Doesn't this prove there's a virus in the cell culture?
Yeah, that's good. I mean, this is the standard question of the virologist or argument. But, you know, they just point these arrows towards particles in their pictures, and we don't know what these actually are because nobody has characterized them, nobody has taken them out and analyzed them, nobody has done transmission experiments with those pieces. It's just a picture out of a cell culture, and that's it. You could find these particles in every cell culture. This is what I am now making an assumption. I say that these particles can be found in every monkey kidney cell culture, which is stressed in terms of adding antibiotics, removing nutrition, like decreasing fetal bovine serum and so on. You can create these particles. What we don't know what they are. They could be artifacts. They could be, you know, spore-like things, exosomes, whatever. There are 1,000 different theories, but it doesn't matter. We don't know what they are because nobody has taken them out and analyzed them. So it's like taking a picture out of the universe and pointing to a star and saying that star is full of viruses. Whatever, it's totally bullshit. Doesn't make sense.
You know, just to emphasize that point, I don't know if you've seen this. I have this little cat that I really like called Pumpkin, and I taught him to say yes or no, and I showed him pictures of published pictures of electron microscopes and asked him which ones were SARS-CoV-2 and which ones were kidney biopsies from 20 years ago or lung cancer. And Pumpkin got 6 out of 11 right, mostly because he likes to say yes. But here are the pictures. So there's the arrow. Is this kidney biopsy from the 90s or SARS-CoV-2? You don't know. How about that one? Or this one? Or this one? SARS-CoV-2 or kidney biopsies or lung cancer, right? You can't tell. Or this one. Looks like, I happen to know this one is kidney biopsy. And on, and you can see all these pictures that look exactly like SARS-CoV-2 we're told, but turn out to be from kidney biopsies or lung cancer specimens before 2000s, there's no chance they're, were SARS.
I think for the viewers, it is just important to know that you can take these pictures from every cell culture, no matter if you add a sample, if you add no sample, if you add a sample from a sick person, if you add a sample from a healthy person, it doesn't really matter. You can always stress the cell culture in a way and always add the ingredients you need to create those pictures. It's totally independent from a virus or whatever. It doesn't really matter. This is important to know.
Okay, next question. Why isn't and you touched on this before, but why isn't the viral sequence the proof of the existence of the virus? And you say they don't do controls. Can we look at this study and see whether they actually did controls?
Well, I can say from like 100% purity, like I am 100% sure that no virologist has ever published a control for a genome sequence assembly. They never control this step. And this is also something I got for my court case. I filed a Freedom of Information Act from the University of Melbourne, from the Doherty Institute, which is, they published one of the first studies of SARS-CoV-2 out of China. And this is a question that I asked them is if they performed controls for the genome sequencing method. So have they performed sequencing with a negative sample, or have they performed sequencing with another reference genome, for instance, measles or HIV or whatever, or any sort of controls, and none of them have performed it, and they even admit that, all of them admit that. So there is no.
What would a control be for a genome sequence? Can you flesh that out for people? So here's the viral genome sequencing, and when you read this, we combine, there's no mention of a control in this.
Exactly. You would need to do exactly these steps that are mentioned here from a negative control.
What does that mean exactly? Walk us through how you would do that.
Okay, you would first take a sample from a healthy person. Okay, the ideal world would be you would take out the virus from the sample and give it to the further steps, but obviously that is not possible. So the best we can do is take a sample from a healthy person or from a person that we say is healthy and has no virus. Then we culture it like the virologists do, and then we run the sequencing and we perform the steps here. So we basically receive from the sequencing all the little parts, the RNA sequences, and then we assemble them in the same way that is performed with the first experiment. So we use the same template. We use the Wuhan template. In that case, they use this one. We use the same chemicals. We use the same parameters in the software in Megahit or Trinity are the names of these tools that have been used. So we use all the same, and we try to find the same solution, so to say. We try to find the same sequence. And in the case that we cannot find it using the same steps, then we can say that the control did not falsify the positive culture, but they are not doing that at all. So they are just sequencing the positive sample, the positive culture.
Let me see if I got that right, so that we can all understand that. So they take the so at no point then, having done the culture, did they has the existence of the virus thus far been proven, right? We know that. So then they take the results of the culture and mix it with some chemicals, produce a RNA library, which is little pieces of RNA, and then they assemble those into the genome, which is based on aligning that to a predetermined template that they told the computer to align it according to this template.
Yeah, and the template is also a computer alignment.
Right.
So that's also important. Yeah.
And what you're saying is anybody who claims to be scientific would do the same thing with what I would say is the following samples. One, with nothing added to the culture. Then something added that's from a healthy person. Then something added from somebody who has, say, influenza or measles. And then the fluid from somebody with, say, lung cancer.
Yeah.
And maybe some other ones. I mean, you would try to do every possible control, and then you would do the same steps. You would culture it, use the same chemicals, break it up into pieces, tell the computer to align that or assemble that according to this template. And if in no cases did you get the same assembled genome, that would certainly strengthen your case that it was only from the from the culture, or the culture from the person with COVID.
Yeah, correct. Yeah, very good explained. And also you can change the template as another control. Yeah. You can say, try to align measles, try to align HIV, try to align Zika, whatever. So SARS-1. So if you are successful in reproducing other templates in the alignment process, that of course also falsifies or brings the virologist in a big issue, a big problem if you get this. Yeah.
And what you're saying, as far as you know, and it sounds like you know, and this is the same conclusion that I've come to, and Andy's come to, and Mark has come to, and Stefan and Mike Stone and Mike Donio and a bunch of other people, Stefano, Kevin, is that we can't find one published paper where they did any of those controls, let alone all of them.
Yeah, and this is a very, very important point, maybe the most important point, because why? For the cytopathic effect, there is some disagreement. There are some virologists saying, oh, we have the mock controls and there were no cytopathic effects. And okay, it's kind of a mess. But in this case, in the genome sequencing control, no virologist has ever claimed to have done the control experiments. I have never encountered a virologist claiming that. In all of my FOIs, in all of my requests, I have never encountered a single virologist claiming that. So here, there is no disagreement. Every virologist agrees that nobody has published a valid scientific control for the genome sequencing methodology. There is one case, I think you did it in a podcast lately. There is one scientist from France, Nice, from the University Hospital of Nice, who said that after publication, they did some random controls and they found positive samples through these steps as well. But this is, of course, ridiculous because you have to do it before you publish the paper, not afterwards.
Right.
You know, so. But this already also, of course, supports our case that we are making, because they produced positive, samples, positive results in their negative control. So. Yeah.
So what I've heard when asked, and I would love to hear what you've heard, they say, well, if you do if you try to sequence, which means assembly, from a sample from a lung cancer patient, the PCR probes don't anneal to anything, and so you don't get anything to assemble. And so why should we do that?
Well, the truth is that they are not just, sequencing the sample and, starting to assemble it. The truth is they are adding many steps afterwards. They are running PCR. They are amplifying the material. They add other chemicals to it. They run another PCR. They run very dirty cycles in metagenomic sequencing approaches. So it's not true that if they would treat the original material, the cell culture, from a negative control in the exact same way, running PCR prior to sequencing it and so on, and adding like other material to it, like human lung fluid, whatever, you can certainly find the same results as proven by Stefan in his I think it's the second or third control experiment, where he showed that. And I mean, what the virologist said just from France, is of course proving that this is true, because they found actually the same sequence in the negative control. So I'm not aware of any virologist who really disagrees with that.
Yeah. Well, even if that, my response to that statement, well, we wouldn't get any annealing, we wouldn't get any amplification with our probes, then fine, why don't you publish that? Just do the same steps and show that you don't get anything, and that would be a good control. But they don't do that. And instead they just say, in theory, it won't work, so we're not going to do it.
Yeah, it's unscientific, of course, because it's also the same reason why they are not publishing the exact steps of the mock infection, because they know if they publish the correct step, if they publish the steps, the full documentation, of course, they will not be able to publish the paper claiming they found a virus, because it would just be ridiculous. Everyone would immediately see that the control is invalid.
Right. Okay, next question. Marvin, why isn't RT-PCR proof of the virus? This is the gold standard.
Yeah, so this is like a circular reasoning, right? Because if the RT-PCR, so real-time PCR or reverse transcriptase PCR, is just we have to check how is the PCR set up, who comes up with this protocol. And in the case of COVID, it was the first guy was a German virologist, Christian Drosten, from the University Clinic Hospital of Berlin. And he basically had no patient sample. He even admits that in his paper. He had no patient sample of COVID, or what is thought of COVID. He had no virus isolates whatsoever. And he just ran sequencing algorithm from the SARS-CoV-1 data that he downloaded as of January 2020. So, and came up with a proposal what this new virus could look like. And then he synthesized the primers based on the result he got from SARS-CoV-1 data, and sent those primers, which are basically the detectors in the PCR. He sent them to China and told the guys in China, oh, check if you find some positivity with my primers. And of course they did, because you always can produce positive results if you do enough cycles. Everyone knows that, even the virologists admit that. So it is not proof of a virus, because you just find 4 or 3 RNA pieces, and the virus is supposed to be a chain of 30,000 pieces. So you just find 4 of them, and if you find them, it's not proof that you have the 29,997 other pieces, right? I think everyone would agree. The only way that the PCR would be a proof of a virus is, of course, if you had run transmission experiments, contagion experiments, and isolated the virus in the first place, and then try to calibrate a PCR, find the correct primers, which would only exist in this viral sequence, which would be specific to that virus and would be found in no other place in the whole universe, in whole nature. Right. They would be specific to the virus, and nobody has done that, of course. So the PCR is invalid proof. It's not validated. It's just and you only, even in theory, even if you say the virus exists, the PCR would just find like 1%, not less than 1%. And that's not sufficient. You need to find the whole thing.
Yeah, I think the principle of that you're talking about is, and as far as I know, this principle exists for everything there is. You cannot say a piece of something comes from a whole unless you've had the whole first.
And unless you prove that the piece is specific to the whole. Yeah. For instance, if we find like a leaf of a tree, if we can prove that this leaf only belongs to one certain type of tree, we can say for sure, if we find a leaf, that it is only from this type of tree or from this family. However, if we find this leaf on another tree, on another type of tree, it's not any more specific. And this is the case for the RNA sequences that they find. Nobody has ever proven that they are specific to a pathogenic virus.
Right.
It's just an assumption.
Interestingly, you might be able to do that with a leaf, but we're talking about a piece of DNA maybe from the leaf.
Exactly.
And that's the same probably with oaks and birches and the rest of it. So the whole leaf might look different, but the pieces don't. And the only way to sort that out is to do a control. Okay, now I'm going to finish with the two tough questions. Let me see if I can ask this clearly. When virologists are making vaccines or propagating, let's say a measles virus, they do, let's say, hundreds of cell culture experiments, one after another, and they say they find the measles virus in every serial culture they do, because they take something from measles, grow it, take that, grow it, etc., on for hundreds, even thousands of times. And they say that if you did that with, say, influenza virus, that the culture would be different, and therefore that proves that you're culturing the virus. Is that true or not? Is that clear what I asked?
So let me get this. Let me repeat if I correctly understand your question. They are saying that when they sequence sample from measles in the culture, it looks different as if they would sequence sample from an influenza patient in after culturing it. Is that correct?
Not necessarily sequence. They say they can tell the difference. They can tell that this is a measles line. And then they even put that in a vaccine, and they say this is a measles vaccine. That's after hundreds or thousands of sequential cultures, whereas hundreds or thousands of sequential influenza cultures, they say gives you a different result.
Okay, that could be, but it's still not a proof that there is a virus. Of course, if I have a sample, like in all of this, there must be a first step. And the first step is some sort of like patient sample. Of course, it can be that there is a difference from one person to the other in a sequential result of culturing it. This can be, but it's still not a proof that it is like a disease-causing virus. for that, you need to perform contagion experiments, transmission experiments. You need to show that the independent variable, which could only be the single isolated purified virus, is causing disease and so on. That would be the proof. But just showing that the line looks different under the electron micrograph or even after sequencing it is not a proof. Of course, I can create different lines by adding different samples from different persons or even treating them in a different way. Of course, it's easy.
Right. They say, though, that just to follow my what do you call it? Trying to debunk this. They say whatever was in the original sample is now so diluted after 1,000 passages that the only thing that could have been carried on is this replicated virus.
Well, in that case, I would check, first of all, what are the chemicals they are using? Are they exactly the same? Are they exactly the same in the measles cultures? Are they exactly the same in the influenza cultures? Then I would check, is the cell line the same? Did they use Vero E6 cells or other cells? Did they use human lung cells? What is the basis of this culture? What are the ingredients? What is the temperature used? What is the CO2 used? Which antibiotics did they use? And so on. How many % of fetal bovine serum? And so on. So if all of these parameters were exactly the same, which is impossible to prove, of course, but let's say they have this documentation and can prove it, then I would start to think, okay, how is this possible? I would deep dive into it. I would do more investigation. It would be very interesting to me. But they don't have this documentation, and if they have it, of course, it's not the same. They cannot. I mean, I'm 100% sure that the influenza culture in that case would have other ingredients or would be treated slightly different under different conditions. And of course, the original patient sample is different also. Okay, they say that it dilutes over time, but it's an assumption. They would need to prove that as well, that it dilutes over time. So it's just fully based on assumptions.
Got it. You know, I've asked a number of people that question, and you got it exactly right as far as I can see, because I have looked into it, and what you find, they say, no, we did cultures. And when you ask exactly that question, were the culture conditions the same for me? Well, of course not, because measles only grows in, WHO cells, and influenza has to grow in Vero cells. So that's, but they're the same but that's absolutely not true. They change the conditions over and over again, and that invalidates the entire conclusion.
Yeah,
okay.
Exactly.
Okay, final question. If what you're saying, and Stefan, is it's the conditions of the culture produce the cytopathic effect, right? Why is a culture ever negative?
Because you need to stress the culture in the right way to kill it. If you don't succeed to kill it, then of course it is fine. It survives for some time. I mean, eventually every cell culture would disintegrate, would show these effects. The question is, at what day? Is it after 6 days? Is it after 2 days? Is it after 8 days? And you can stress the culture in the right way to do it after 8 days or to do it already after 3 days. Or it just depends on how you stress it. And I'm not aware of a cell culture, I mean, that survives forever, right? So eventually every cell culture shows
they have to replenish
signs of degeneration, right? Of course, because you need to maintain also the nutritional medium. You need to maintain the nutrition. You need to maintain the temperature. You need to pass on, you need to take off the supernatant and pass it on regularly. Otherwise, the cell culture would just grow too much and so on. So this is a highly controlled process over time. It is a very unnatural environment. It's a lab experiment, which is very, very, very sensitive. So of course it's totally like manipulatable. The lab scientist can do whatever he wants with the cell culture. He can create CPE after one day. He can create CPE after 1 week. It is up to him, right?
Yeah.
So.
Right. And when you look into, so they took lung cancer and they looked for viruses and they say there's no CPE, and you find that they didn't stress the cultures in the same way as they do with the so-called viral cultures. So
yeah, there is also an important point here. If you take lung cell lines, the stress that you need to add in terms of chemicals, in terms of temperature and other variables, parameters, might be different than in monkey kidney cells, for instance. So every cell culture has a different, capacity to survive or to stay alive when stressed. Some need streptomycin, some need penicillin, some need a mix of both, some need 5% or even less, 1% fetal bovine syndrome to show these cytopathic effects. It differs from the tissue which you take. Some tissue is very, very resistant, and some tissue is very, very weak and disintegrates immediately. And you will never succeed to keep it alive for 2 days. Some tissue is just impossible to run these experiments on. That's why they decided so many years ago to take the Vero E6 cells, because it's so nice. They can so greatly manipulate them. They can let them living for 1 week. They can kill them after 3 days. It's perfectly for the virologist to play. It's the perfect playground.
Right. And unless you do control every step of the way, you never know which parameter actually caused the death.
Yes.
And yeah.
You need in the controls, the goal should always be to only change one single variable.
Right.
And the scientific control should be designed in a way that allows that. So even the mock infection, if it existed, is not a good control, as we said earlier, because adding no sample at all is changing already too many variables. It's not a good control.
Right. Okay. I think we got it. I don't know of any other questions. I think at this point, I can say, for anybody listening who's still on the fence, like, who's right here? These people they say they have controls. I would say just ask yourself the following question. If you are a scientist, why wouldn't you take every opportunity to do every possible control you could think of? Or your plumber friend, or your chemist friend, or anybody else you could imagine to do, well, what about if I just put, nothing, or lung cancer, or something else? Why wouldn't I do every possible control? And if the answer is stonewalling, meaning, oh no, we're not doing controls, we don't need to, that should make everybody suspicious that there's something really wrong in the house of virology. And that's what I think we're trying to bring to light. And so people listening can ask themselves, when they're stuck on these, they look at it and they see the control name, why wouldn't they do this? Why wouldn't they want to prove this in every way possible? And I think that gives you the answer right there.
Exactly. Yeah. And also, I can show this very quickly. People will be able to see it also in interview with Sam and others, but just because it is now perfectly matching what you said. I even asked the University of Melbourne this question: why are you not doing the controls? What is the reason? Why are you not doing it? And they say at the time there was no established method for sequencing SARS-CoV-2. In order to maximize the number of sequencing reads, we elected to dedicate an entire flow cell to the positive culture sample to maximize our recovery of the viral reads, and not spend reads on sequencing negative samples. So basically their excuses, they had capacity limits and they just run the control, they didn't run controls, they just performed the experiments on the viral reads, as they say. But here you can see it's a totally unscientific answer. They are not giving a good reason. For instance, oh, these controls were performed before, or we already validated our method, check this study, this citation. No, they just say we had no capacity to do it. And of course, if you get this answer, is totally clear here what is going on. And of course, they admitted, like I said, even before, that they did no controls for other viral genomes in the assembly. The assembly was performed only against the release Wuhan 1 reference. And also, they said that sequencing de novo assembly and de novo assembly was only performed on positive infected material. So they admit no control was performed on the negative control. So, I mean, it cannot be any clearer than this. I mean, this is just perfect. Everyone showing this in the court, I'm not sure that the judge could go along, could find escape holes. It's just impossible.
In other words, this is clearly saying we didn't control for the genome assembly, so we don't know if you would get the same result if you put lung cancer in there or nothing in there or a well person. We didn't sequence it again. We didn't use a template from another virus, so-called. So we have no idea if that's what we would get if we did our assembly.
Correct. Yeah, it's just not scientific, and the reasoning they give to not run these experiments is just ridiculous. I mean, this answer, I'm quite.
I mean, after
surprised.
all, we only have $100 billion to spend. Why should we spend $1,000 on doing so?
I'm so surprised that I received this honest answer. It's incredible. I would never have thought to get this answer. I'm really surprised.
Great. All right. Anything else you want to share with us, Marvin? Or I think we got it here.
Yeah, no, I think the case is very clear. I will keep you and of course the community updated regarding the court case. I will, call the court once every week and check if the judge is, healthy again and if there is a new appointment. So far, I did not receive any new appointment. I hope that it will be soon. The lady at the telephone is very, very friendly, and she told me that there will be probably a new appointment sent to me in November, which should be in the next weeks. But let's see. Yeah. My gut feeling is that there will be no appointment. Actually, they will just close the case. But let's see. Let's hope for the best.
Great. All right, Marvin, keep in touch. And like I say, if you get further and you need some help, let us know, because I'm pretty sure my people will step up for you. And I really appreciate it. You were wonderful to talk to you, and keep in touch, please.
All right. Thank you, Tom.
Okay. Thanks, Marvin.